Once upon a time, there was a ship sailing in the vast Pacific Ocean. It was somewhat sunny that day, with the breeze touching the flags on the mast and the waves sparkling. On that ship, there was a pirate and its crew named Stella. It was when the pirate woke up to the noise that Stella made from cleaning the cabin.
Quiet, quiet. I'm starving. Hey, Stella, get me something to eat. I want me some prawns.
The pirate yelled.
Aye, Captain Crook. Will do, sir. But wait, there are so many fishes. Which one of these is rich in protein?
Said Stella.
I don't know, but I hear tunas contain a significant amount of protein.
Really? Okay, hold on. Oh, look at this, Captain. Is this one it?
Suddenly, a tuna flew in from the boat.
Hello, I'm Mary Joy.
Why is it talking?
Shouted the pirate and Stella.
Why do you need proteins, Captain?
Asked Mary Joy, the tuna.
Yeah, why do you need it? What even is a protein?
Asked by Stella, who's now confused.
All right, Stella. I want you to listen to this very carefully.
Proteins are like the unsung heroes of your body. Tiny, powerful, and absolutely essential. They are the architects of our existence. Imagine your body as a puzzle machine. Proteins are the workers, carrying out crucial tasks, constructing and maintaining structures, and ensuring everything runs smoothly. Now, what is a protein? Proteins play a vital role in sustaining and regulating the many processes that define living organisms. Proteins, often referred to as the building blocks of life, stand as the most abundant organic molecules in living systems. Proteins are large, complex molecules that play many essential roles in the functioning of the human body. At the heart of every protein lies the amino acid, the fundamental building block that gives rise to the remarkable diversity of these biomolecules. Composed primarily of carbon, hydrogen, oxygen, and nitrogen, collectively known by the acronym TRON, proteins exhibit remarkable diversity due to the varied proportions and sequences of the 20 amino acids present within the protein molecule. The multitude of permutations and combinations of these amino acids is the driving force behind the boundless array of proteins observed in nature. The different permutation and combination on the arrangement of amino acids significantly affect the protein structure and function. Think of it as our famous Filipino childhood game, Dr. Quack Quack, where players can rearrange themselves in many ways, making it difficult for the doctor to dismantle them. Amino acids can also have different arrangements, and with each distinct arrangement lies a unique structure and function. Each amino acid shares a foundational structure, comprising an amino group, a carboxyl group, and a distinctive hydrocarbon side chain. The connecting force within these chains is the peptide bond, an amide linkage that joins the amino group of one amino acid to the carboxyl group of another. This bond, while seemingly simple, serves as the molecular glue that holds the amino acids in precise order. There are several forms of peptide bonds, beginning with dipeptides, consisting of only two amino acids and a single peptide bond, and extends to tripeptides, comprising of two, or in some occasions, three peptide bonds, then oligopeptides, which contain 3 to 10 peptide bonds. For longer chains exceeding 10 amino acids, the term polypeptide is employed. These polypeptide chains serve as the backbone of proteins, acting as a framework upon which the complex structure of these molecules is established. Proteins can create extensive polypeptide chains. Beyond their linear nature, these polypeptide chains exhibit a specific ability. They can fold and bond with themselves to create three-dimensional protein structures. This folding process is not natural. Rather, it is a meticulous choreography that results in the formation of a complete functional protein. Furthermore, individual proteins can often join forces to form more complex assemblies. These conglomerates of proteins work collaboratively, each contributing its unique function to achieve a higher order biological purpose. The shape of proteins, like a blueprint, dictates their specific function in the intricate machinery of life.
Now, let's move on to the dynamic role of proteins in pivotal cellular functions, its intricate mechanisms governing cell division, cell membrane, and cell metabolism. To keep the cell cycle on track, there are cyclin-dependent kinase, or CDKs in short, which are protein kinases characterized by needing a separate subunit, a cyclin, that provides domains essential for enzymatic activity. Together, these CDKs regulate cell division and even modulate transcription in response to several extra and intracellular cues. In addition, the mitotic spindle-associated protein PRC1 is known to be essential for cytokinesis, and recent studies have shown that one of its functions, namely its ability to bind to and bundle microtubules, is required for central spindle formation during the anaphase. Proteins are also essential for the metabolism of the cell, especially through the molecule called enzymes. These enzymes are like catalysts made of proteins that speed up biochemical reactions by helping molecules rearrange themselves. Because of this enzymatic action, cells can adjust to different environments and control their own metabolic pathways, which are basically the routes for different chemical reactions, therefore making it crucial for a cell's survival. Whereas the lipid bilayer determines the basic structure of cell membranes, proteins are responsible for most membrane functions, serving as specific receptors, enzymes, transport proteins, and so on. Membrane protein is a protein molecule that exhibits diverse functionalities, facilitating the passage of hydrophilic molecules across the cell membrane. Let us now dive into the advantages of consuming proteins, how it affects our body, and serve its benefits as the building blocks of amino acids. First, proteins help the body repair itself after injury. Our body needs protein to help build and repair muscle, skin, and other body tissues. Proteins provide the main blocks of our tissue growth, cell renewal, and repair throughout the wound healing process. Then, it also helps maintain weight loss. Many people who increase their protein intake tend to lose weight almost instantly. Protein helps us maintain our weight loss because a high protein diet boosts metabolism and leads to an automatic reduction in calorie intake and cravings. Next, proteins lower the blood pressure. Recent observational studies and clinical trials have suggested that increased protein intake, particularly protein from plant sources, might actually reduce blood pressure and prevent cardiovascular disease. Furthermore, it increases muscle mass and strength. Protein increases our muscle mass and strength as it is the building block of our muscles. Therefore, eating adequate amounts of proteins help us maintain our muscle mass and promotes muscle growth whenever we are working out. Also, proteins boost bone health. A higher protein diet increases insulin-like growth factor 1, which is a key mediator of bone health. To further understand our discussion, proteins also perform many functions as seen on the table, highlighting their crucial contributions to various aspects of our biological existence. So first, actin-myosin filaments are two protein molecules in the muscle which are used for movement as the muscle contraction results from an interaction between both of them. On the structure, one of its examples is the collagen. Collagen is a major structural protein and it is also the most abundant protein in the human body, forming molecular cables that strengthen the tendons and resilient sheets that support the skin and internal organs. And then, hemoglobin demonstrates the protein's transportation function since the hemoglobin in red blood cells, an example of globular protein, transports oxygen through our bloodstream. Casein is a protein found in milk for use by some newborn animals as it is rich in calcium. Proteins function as a defense through the antibodies, as it is a protein produced by the body's immune system when it detects harmful substances, called antigens, which help animals fight off invasion by viruses and bacteria. Lastly, proteins regulation function. Enzymes are proteins composed of amino acids linked together which helps control chemical reactions in the body.
Proteins are crucial molecules for life, and despite seeming simple, they have a complexity that requires analyzing their structure. This involves looking at both the primary structure and the more detailed further structures that determine how they work. But before anything else, let us proceed to our experiment entitled Denaturing Proteins. And now, for us to gain a greater understanding of the structure of proteins, we will be conducting a simple experiment entitled Denaturing Proteins.
The materials that we use for this experiment are 4 pieces of plastic cups, 4 pieces of 1/4 egg yolk, 4 pieces of stirrers, rubbing alcohol, hot water, tap water, timer, and measuring cup. First, pour 1/3 cup rubbing alcohol into one container, room temperature water into another, and the rest of the water into another container. Next, quickly put 1/4 of the egg yolk into the boiling water, then put 1/4 into the alcohol, 1/4 into the room temperature water, and the remainder into the final empty container. Third, observe any immediate changes that occur in the terms of egg white color and consistency. Then, wait for 30 minutes. Last, use a stirrer to inspect the state of the egg whites in each treatment and note how they may have changed over time.
Now, let's move on to the results of the experiment. Transformation of denatured egg white from clear to white is a result of alterations in protein structure induced by various external treatments. In the control group where the egg whites were left at room temperature, the proteins maintained their original shape, keeping the solution clear, as you can see. On the other hand, cooking it with hot water led to denaturation, as the heat broke hydrogen bonds, causing proteins to unfold and the egg white to turn white and rubbery. Alcohol, on the other hand, acting as a bond disruptor, denatured proteins similarly to heat, but at a different rate, as it had to diffuse into the egg to affect proteins. Lastly, tap water also denatured egg white by breaking bonds, but its impact was milder compared to alcohol or hot water. Now, there you have it, the result of the protein denaturation. But that's not it. I want to ask you a question. What is the point of this experiment? Why do we need to conduct this protein denaturation to understand the structure of proteins? Well, protein denaturation is crucial for understanding the diverse structures and functions of proteins. By subjecting proteins to various denaturing agents, such as heat or chemicals, scientists can unravel the intricate 3D structures of proteins and explore how these structures relate to their functions. Now, let's move on to the different structures of proteins. The primary structure of a protein, although it might seem basic since it's considered as the simplest of protein structure, is like the cornerstone of its whole structure. It involves a straight order of sequence of amino acids in a chain, and this order plays a key role in deciding the protein's special identity, structure, and job. The amino acid sequence, known as the primary structure of a protein, is responsible for the folding and internal bonding of the straight chain of amino acids. This process ultimately shapes the protein in distinctive three-dimensional form. On the other hand, secondary protein structures refer to specific folded arrangements that occur at a local level within a polypeptide chain. While the primary structure outlines the linear sequence of amino acids, the secondary structure delves into the local patterns of folding that shape portions of the chain. The alpha helix is a distinctive and common structural motif in proteins, characterized by its spiral or helical shape. This secondary protein structure is held together by hydrogen bonds, which are interactions between specific atoms within the amino acid chain. In the alpha helix, hydrogen bonds forms between the oxygen atom in the carbonyl group of one amino acid and the hydrogen atom in the amino group of another amino acid. Now, I want you to look at this alpha helix model made from aluminum wire and yarn. As the alpha helix progresses, it completes one full turn, approximately 3.6 amino residues, as you move along the alpha helix structure. The helix completes a full circular turn about its central axis, roughly every 3.6 amino acid building blocks in the protein chain. This specific ratio of residues per turn is a critical feature of the alpha helix and contributes to its uniform and repetitive structure. Beta-pleated sheets represent another common secondary structure in proteins, distinct from the helical structure of alpha helices. These sheets are characterized by a flat, extended arrangement of the protein chain. These sheets can either be parallel or anti-parallel, referring to the orientation of adjacent strands within the sheets. The repeating pattern of hydrogen bonds results in a pleated appearance, with alternating rise and fall of the strands. This arrangement allows for an extended or planar structure, making beta sheets well suited for forming a core of many protein structures.
In the tertiary structure of the protein, we can now state the further folding of the secondary structure into an overall folding of the polypeptide chains. Within this structure, two prominent molecular shapes emerge, the fibrous having elongated rope-like structures that are strong and hydrophobic, and the globular protein, which generally have a more compact, spherical, and hydrophilic structure. This folding process is upheld by various stabilizing forces such as the hydrophobic interactions, hydrogen bonding, ionic bonding, disulfide bridges, and the van der Waals forces. For the last structure of protein, the quaternary protein structure. It is formed when multiple polypeptide chains link together to form a functioning unit. Each polypeptide chain is referred to as a subunit. Proteins with this type of structure may consist of more than one of the same or even different types of obtained subunit. At this point, the protein is fully functional and able to perform its specific roles within the body. One of the examples of a fully functional protein is the hemoglobin. It is an iron-containing protein that binds oxygen molecules and contains 4 subunits, 2 alpha subunits and 2 beta subunits.
Oh, so that's what a protein is. Now, I know what a protein is, its function, its different structures, as well as its importance in the human body.
Stella said.
Yes, but the protein that I possess is classified differently from the others.
Mary Joy added.
That's right, Stella. There are numerous classifications and examples of proteins.
And every one of these classifications vary in structure, composition, and function.
Oh, really? Can you help me out? I want to know more about proteins.
Said Stella, who's now begging to learn more.
Okay, listen to this.
For the classifications of protein, they are divided into 3 groups which are categorized based on structure, composition, and function. To clearly understand this, let's go discuss each one at a time. If we're going to classify protein based on molecular shape, it is further classified into 3 distinctive types of fibrous, globular, and intermediate protein. Starting with fibrous protein, wherein it occurs when the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, forming long fibers or sheets. Such proteins are linear in shape and are very tough and strong. Generally, proteins like this are insoluble in water, which means that they are hydrophobic or scared of water, at least not literally. The most significant functional structure of fibrous proteins is their secondary structure and lack of tertiary structures. Fibrous proteins also have its main duty, which is to perform the structural functions that give strength and protect cells and tissue. Example of this is collagen, which gives tissues and cells structural integrity and support that helps shield them by creating a robust framework that withstands mechanical stress and preserves the structural integrity of different bodily parts. Other examples of fibrous proteins includes keratin, which is presented in hair, wool, and silk, myosin, which is presented in muscles, and collagen, which is presented in silk, tendons, and ligaments. Next one is globular protein, which is the result when the chains of polypeptides coil around to give a spherical shape. These proteins are softer than fibrous proteins, and unlike those, they are hydrophilic or soluble in water. Moreover, tertiary structure is the most crucial functional structure of it, representing overall folding of the polypeptide chain in its structure. Their main function is to synthesize enzymes, antibodies, and hormones because of their compact folded structures that enable certain interactions with substrates or receptors, facilitating immune response, catalytic activity, and cellular signaling regulation. Globular proteins are also the majority of proteins found in cells due to their adaptable shapes, which allows for processes like enzymatic activity, solubility, transport, control, and structural support. Examples of globular proteins include insulin, which is primarily found in pancreas, and albumins in bloodstreams. Last but not the least is the intermediate protein. From the name itself, intermediate, this classification of protein is a mixture of both fibrous and globular protein. These proteins have a short, roughly linear structure and are also hydrophilic. In addition to their solubility in water, intermediate proteins also plays a vital role in blood clotting, contributing to the coagulation process, which involves a series of enzyme activities that leads to the production of a blood clot to stop a bleeding. An example of an intermediate protein is fibrinogen, which of course plays a pivotal role in forming blood clots and neurofilament. Now that we've already discussed the classification of proteins based on structure, let's then tackle its classification based on its composition, in
which it involves a classification between simple and conjugated protein. Starting with simple proteins. Their structure is characterized as simple and straightforward due to their composition being solely based on amino acid joined by peptide bonds. These are also proteins that are not attached to any other elements, in contrast to more complex proteins that contains more components or complex folding patterns. Moreover, enzymes that break down simple proteins only produce amino acids as byproducts elements, indicating that the most basic protein of all is the one that belongs in the simple protein category. Examples of this are prolamins, globulin, glutenins, and albumins.
So the last broad type of protein according to its composition is conjugated proteins. Conjugated proteins are complex proteins. When hydrolyzed, it releases one or more amino acids, inorganic and organic components. Conjugated proteins are usually globular in shape and are soluble in water. They are tightly or loosely bound to one or more non-protein parts. These non-protein parts are called prosthetic groups. Prosthetic groups are essential in the biological function of these proteins. Prosthetic groups can be metal ions, nucleic acid, and phosphoric acids. Lipoproteins are proteins conjugated with a protein shell over a lipid core that contains cholesterol, triglycerides, or both. Let's now proceed to 8 types of protein based on its function. Structural proteins maintain cell shape, like akin to a skeleton, and decompose structural elements in connective tissues, like cartilage and bone in vertebrates. Most of them are fibrous proteins and are insoluble in water. Here's an example of structural protein, elastin. Just like a rubber band, it is stretchy, but elastin is stretchy protein and located in tissues and organs of higher vertebrates, making it insoluble in water. Let's now move forward to the second function, enzymes. Enzymes are called biological catalysts because they speed up the rate of biological reaction in the body without getting used up or altered in the process. These are the proteins that act upon substrate molecules and decrease the activation energy necessary for a chemical reaction to occur by stabilizing the transition state. An example of an enzyme is maltase. Maltase is an enzyme found in plants, bacteria, yeast, humans, and other vertebrates. It catalyzes the hydrolysis of maltose into 2-glucose sugars. Hormones are signaling molecules, often proteins or steroids, released by endocrine glands to regulate physiological processes like growth, metabolism, and reproduction. For instance, insulin controls blood glucose levels. Steroid hormones are a group of hormones derived from cholesterol that act as chemical messengers in the body. The steroid hormones regulate many physiologic processes, including the development and function of the reproductive system.
Respiratory pigment is a colored metalloprotein that serves a variety of important functions, its main being oxygen transport. Other functions performed include oxygen storage, carbon dioxide transport, and transportation of substances other than the respiratory gases. They are colored proteins and they contain pigments called chrome as their prosthetic group. One example is myoglobin, which is a protein that's found in your striated muscles, which includes skeletal muscles and heart muscles. Its main function is to supply oxygen to the cells in your muscles called myocytes. Transport proteins are located in the cell membrane of all cells and are responsible for moving materials into and out of the cell. Transport proteins can also be found within organelle membranes inside eukaryotic cells that are found within the membrane itself, where they form a channel or a carrying mechanism to allow their substrate to pass from one side to the other. The substances transported by these proteins can include ions such as sodium and potassium, sugars such as glucose, proteins and messenger molecules, and many more. Transport proteins generally perform 2 types of transport. Facilitated diffusion, where a transport protein simply creates an opening for a substance to diffuse down its concentration gradient. And active transport, where the cell expends energy in order to move a substance against its concentration gradient. An example would be serum albumin, which is the largest protein component of human blood, and is an important factor in the regulation of plasma volume and tissue fluid balance. Contractile proteins, on the other hand, are proteins that aid in the contraction of muscles. These proteins are involved in localized contractile events in the cytoplasm, motile activity, and cell aggregation phenomena in the latter. Contractile proteins are proteins that mediate sliding of contractile fibers contraction of a cell's cytoskeleton, and of cardiac and skeletal muscle. One example would be actin, which is essential for a tremendous range of cell functions. A partial list includes cell division, migration, junction formation, raw material modeling, transcriptional regulation, vesicle trafficking, and cell shape regulation. Storage proteins serve as reserves of metal ions and amino acids, which can be mobilized and utilized for the maintenance and growth of organisms. They are particularly prevalent in plant seeds, egg whites, and milk. It is a type of protein that is stored inside cells or tissues as food and can be assembled when needed to provide energy. This type of protein stores amino acids for the body until it is ready for use. Many storage proteins store amino acids in embryos and developing organisms. These types of proteins are stored in a special structure called the protein body. It is located in the cotyledons and endosperm of the seed. One example would be casein, which is a protein found in milk that gives milk its white color. Cow's milk consists of around 80% casein protein. Moving on, a toxin is often considered a specific type of poison, a poisonous substance produced within living cells or organisms. It is a naturally occurring organic poison produced by metabolic activities of living cells or organisms, which occurs especially as proteins. One of the main ways they damage your body is by poisoning your enzymes, preventing your body from functioning properly. Your body relies on enzymes for every physiological function. When toxins damage your enzymes, they prevent the production of hemoglobin in your blood, which can accelerate aging. It also can lead to the failure of energy production and lower your protection against oxidative stress. One example would be disintegrates, which are small cysteine-rich proteins found in a variety of snake venom. These proteins selectively modulate integrin function, heterodimeric receptors involved in cell-to-cell and cell-to-matrix interaction.
Oh, that sure is a lot to process. But I had learned so many things today. Anyway, I'm gonna have to catch Mary Joy for you, Captain. 1, 2, 3. Oh no! I forgot! We don't have a fishing rod.
Yes!
Hey! Catch it! It's getting away! Catch it! Catch it!